In the adult torso, the kidneys and reproductive organs appear entirely distinct. One system is dedicated to high-pressure filtration and drainage, while the other is specialized for reproduction, positioned separately within the pelvic cavity. Despite this physical separation, they are clinically inseparable. When a newborn is diagnosed with a congenital kidney defect, physicians immediately screen the infant for anomalies in the reproductive organs. If one system fails to form correctly, the other often follows. This happens because these two systems, despite their different roles, are vulnerable to the same structural errors. Their connection is found in their origin. In the early embryo, they do not develop as separate entities. They evolve from a single shared source of tissue. By week 3, the embryonic disc has 3 germ layers: ectoderm, mesoderm, and endoderm. Isolating the mesoderm reveals longitudinal strips. The central glowing strip is the intermediate mesoderm. It soon bulges outward, forming the urogenital ridge. Every filter, collecting duct, and reproductive organ in the adult human body traces its physical existence back to this single, unspecialized ridge of embryonic tissue. To build these structures, the urogenital ridge divides into 2 parallel tracts: the nephrogenic cord for the urinary system and the genital ridge for the gonads. The urinary system develops along the nephrogenic cord in 3 successive overlapping stages, moving from the top of the cord down to the pelvis. The first stage is the pronephros. Appearing in the cervical region by week 4, it is a primitive cluster of non-functional tubules that disappears almost as soon as it forms. The second stage is the mesonephros. Forming in the thoracic and lumbar regions, this structure filters waste for a short period. It connects to a major drainage pipe called the mesonephric or Wolffian duct. By the 8th week, the filtering tubules of the mesonephros dissolve, but the long Wolffian duct remains behind. It will be repurposed for the reproductive tract. The third and final stage is the metanephros. Beginning in the 5th week at the bottom of the cord, this becomes the permanent kidney. Construction of the metanephros requires 2 distinct tissues. A smooth branch called the ureteric bud sprouts from the base of the Wolffian duct and pushes into a dense cluster of intermediate mesoderm called the metanephregenic plastema. When they meet, they initiate reciprocal induction. The branch secretes chemicals that keep the tissue cap alive, while the tissue cap secretes signals that force the branch to divide. The branching ureteric bud creates the internal plumbing, the ureter and collecting ducts. The surrounding blastema tissue differentiates into the filtering units, the nephrons. The kidney is a negotiated structure. If either tissue fails to signal the other, the entire process halts and the embryo develops with 0 kidneys on that side. As the kidneys form, the main drainage chamber in the embryonic pelvis must be divided to accommodate the growing digestive and urinary tracts. This cross-section shows the cloaca. In the early embryo, this single chamber receives waste from both the hindgut and the urinary tract. During the 7th week, a partition called the urorectal septum grows downward, splitting the chamber into 2 separate compartments. The back compartment becomes the anorectal canal, while the front compartment, the urogenital sinus, becomes the urinary bladder and urethra. The growing bladder wall must absorb the ends of the kidney tubes and reproductive ducts, pulling them apart so they eventually open into the bladder at separate points. As these tubes integrate, the mesodermal tissue they bring with them forms a smooth triangular patch inside the bladder called the trigone. This patch remains distinct from the rest of the bladder wall, which is derived from endoderm. With the plumbing in place, the kidneys face a mechanical shift. They currently sit within the pelvis, far below their final adult position. By the 9th week, the kidneys are located in the upper lumbar region.
This is not an active climb. Rather, the embryo's lower body elongates, leaving the kidneys positioned high in the abdomen. This development requires constant coordination. These organs must differentiate their tissues, stretch their ductwork, and replumb their connections while the embryo's anatomy rapidly expands around them. We return now to week 7 to follow the second half of the urogenital ridge, the genital ridge. This is the indifferent gonad stage. At this point in development, genetic male and female embryos appear identical. Every embryo possesses a dual system. Flanking the central gonad are two separate sets of tubes, the mesonephric or Wolffian ducts and the paramesonephric or Mullerian ducts. The divergence from this shared state depends on the SRY gene, located on the Y chromosome. On the left, we follow the male pathway. The SRY gene triggers the gonad to become a testis, which immediately begins secreting two specific hormones. The first, anti-Mullerian hormone, causes the female tubes to dissolve. The second, testosterone, stimulates the Wolffian ducts to differentiate into the male reproductive tract, including the epididymis and ductus deferens. On the right, we follow the female pathway. Without the SRY gene, the gonad becomes an ovary. Because no early hormones are produced, development follows a different course. Without testosterone, the male ducts wither. Without anti-Mullerian hormone, the female tubes survive, merging at the midline to form the uterus, cervix, and upper vagina. The body builds a universal dual-tract template, then selectively demolishes half of it based on specific hormonal signals. This same logic dictates the formation of the external genitalia, which remain identical until week 9. This is the indifferent stage for the external genitalia. Every embryo starts with 3 identical building blocks: a central tubercle, inner urogenital folds, and outer labioscrotal swellings. Under the influence of androgens, the male transformation begins. The tubercle elongates, the inner folds fuse to enclose the urethra, and the outer swellings merge to form the scrotum. In the absence of androgens, the same blocks grow without fusing. The tubercle becomes the clitoris, while the inner folds and outer swellings remain separate. The homologies are exact. The penis and the clitoris are formed from the same starting tissue, as are the scrotum and the labia majora. Anatomical dimorphism is not a result of different starting materials. Male and female external organs are the same mesodermal tissue, simply organized into different configurations. In a clinical setting, developmental anomalies follow a strict logic. They occur as mechanical or hormonal interruptions of the specific steps we just mapped out. This model shows a horseshoe kidney, where the 2 kidneys are fused together and trapped beneath the inferior mesenteric artery. This is a mechanical error of early development. If the 2 tissue caps are pushed too close together, in the pelvis, their lower poles fuse. As the body elongates to pull them upward, the fused mass catches on the arterial supply, halting their move to the lumbar region. We see a similar logic in a bicornuate or heart-shaped uterus. This is a timing failure during week 7. The 2 female Mullerian ducts drift toward the midline, but fail to complete their fusion, leaving the top of the organ split in 2. Shared origins also explain persistent Mullerian duct syndrome in genetic males. In these cases, the SRY gene and testosterone successfully build the male tract. However, if the testes fail to produce anti-Mullerian hormone, the female tubes are never dissolved. The male is born with a fully formed uterus and uterine tubes hidden internally. Because the kidneys, internal ducts, and external genitalia all emerge from the same intermediate mesoderm, a disruption to that tissue ripples across both systems. The human body functions as a highly choreographed, 4-dimensional fold. To successfully treat the adult patient, a physician must understand the developmental history that shaped them.